Kia ora. I'm going to speak for the next 15 or 20 minutes about some basic genetics to hopefully get you started on your learning journey to understanding genomic health and medicine. So on this slide, we have some key concepts that are fundamental to genetics and how we describe health conditions. So typically we refer to conditions as being single gene disorders. So this is whereby one gene has been identified as being responsible. So conditions like cystic fibrosis, phenylketonuria, Huntington's are all single gene disorders. We also talk about genes having conditions being the result of chromosomal abnormalities. So often referred to aneuploidy, which is extra chromosome numbers. So things like trisomy 21, 13, and 18 are all compatible with life and are chromosome abnormalities. And the sex chromosome aneuploidies like Klinefelter's, or where there's one less chromosome in the case of Turner syndrome. So when we talk about single gene disorders and chromosome abnormalities, these result from something that happened prior to birth. And in the case of single gene disorders, prior to conception, they were typically carried by the parent unless there were de novo mutations. And so these are, with single gene disorders, they are referred to as germline mutations or variants in a gene that changes its functionality, that determines that gene protein that's made is dysfunctional and then contributes to the disease symptoms and phenotype. Other type of condition that we talk about that is important also for genomic health and medicine, and arguably the most important because it affects the most people globally, are the complex diseases. So these are conditions that have multifactorial inheritance, so multiple genes plus environmental contributions contribute to the manifestation of multifactorial inheritance. On the slide here, we've got a karyotype. So as humans, this is to remind us we have 46 chromosomes, an X and a Y if you're male, two X's if you're female. DNA is where we find all of our genes. The DNA is packaged into chromosomes, which we can see here, and the chromosomes are packaged into the nucleus of the cell. And so every nucleated cell of your body contains exactly the same genetic information. The gametes are the exception because they have half the genetic information. And in that DNA has to be copied absolutely faithfully in every cell, and the DNA code contains the information that for about 20,000 genes, the coding sequence for about 20,000 genes, and they will be specific to each human being. Everyone has the same genes, but in terms of the sequence that makes up variation from person to person, that's what makes us different and unique. So DNA, where is it and what does it do? So as I said, the DNA is packaged into chromosomes and is found in the nucleus of every cell. So we have these 20,000 genes, and they're not being active all the time. We refer to a gene as being expressed when it's active. When a gene is expressed, it is when that DNA is being read by the cellular machinery, translated to make a protein. There are two stages that it goes through. It's just a process of transcription and translation. And essentially, the proteins that the DNA codes for are what make us human. It's what everything that makes us. They're the building blocks of what make muscle, tissue, immunoglobulins, digestive enzymes. Everything that's a protein is made in this process of transcription and translation. And so the code that we contain in the nucleus of every cell codes for those proteins. If we have variants in our DNA, and if those variants are in the coding sequence, then they can have an impact on the protein that's produced, making it dysfunctional, abnormal, or normal functioning protein, or a non-functioning protein. This slide illustrates transcription and translation. These are two key stages in taking that message. So the DNA in our cell, and the nucleus is essentially like an instruction manual to make us as a human being. When we need a protein, the process of transcription and translation reads that genetic code in the double-stranded DNA, transcribes it, the segment for that particular gene that needs to produce its protein, into a piece of messenger RNA, and the messenger RNA leaves the nucleus, goes into the cytoplasm where it's translated into a protein, and then it will be post-translationally modified to make it a functional protein that our body requires. Now, if we have between 20 to 30,000 genes as a human being, and we have all of those contained in every nucleus of every cell of our body, not all of them are switched on or being transcribed and translated at any given time. There are a number of our genes that were only active in embryogenesis or had a particular role in embryogenesis. And the ways in which genes are utilized and when they're switched on and how they're switched on in different cells and different tissues is really important in terms of how we function as a human being. So how do genes work? So this slide here gives you a simplified version of how a gene works. So DNA is double-stranded, and I've put it single-stranded just for ease of understanding. This part here would be the gene body. Essentially, genes are made up of segments of the DNA, typically over a couple of kilobases. And in those segments of DNA, you'll have elements that are called exons, and these are the parts of the DNA that are coding for that gene, and you have introns in between. These are intervening sequences between the exons, and they're the non-coding sequence. In the coding part, or the body of the gene in the exonic sequence, this is the part that gets transcribed into mRNA and then translated into a protein. The first exon always starts with methionine, which is the amino acid at the beginning of every protein, and that's coded for by the DNA sequence ATG and becomes AUG in RNA. So methionine is a signal to the cell that that's the start of the gene. And there are lots of other signals and important elements of recognition sequence to cellular machinery that signify where a gene starts, where a gene ends, where the introns need to be spliced out, where bits need to be added at the end of transcription, and so on. So we have the gene body, which is the exons, and we also have this area here. And when we talk about DNA, we give it direction. It goes from 5' through to 3'. So 5' is kind of to the left-hand side, and this is anything to the left of the start of the gene is talked about as being upstream. So you have an upstream promoter. So the promoter region is the part that's crucial in determining whether this gene is switched on and transcribed and translated. You have the downstream region closer to the 3' end, which is towards the end of the gene, and there'll be sequences there that signify when to stop transcription and translation, where to add a poly-A tail, and elements which enable that piece of mRNA to then be able to move out of the nucleus into the cytoplasm. So we have a promoter region. The promoter region is really important, and when we talk about epigenomics later on, we're going to talk about promoter region and how this area is suppressed or activated to allow genes to be switched on or off. In a simplified world, you have transcription factors, and these are proteins in themselves, or they can be hormones. And these transcription factors are produced in response to particular stimuli. They will bind if there's a cascade, and then they will attach to the promoter region of a gene. When they attach to the promoter region of a gene, this allows the gene to be activated for transcription and translation, which enables this gene's protein to be produced. And that protein then has a function in the body, albeit an enzyme or an immunoglobulin or a protein that's important in structure and function, for example, myosin. These proteins are made until they're no longer required, and this signaling cascade becomes inhibited through the cellular mechanisms. So the switching on and off of genes can be responsive, it can be transient, or there will be some genes that need to be switched on the whole time because they have an important protein product that needs to be produced. If somebody has a genetic mutation, the severity in terms of the disease it causes is determined by the impact it has on the ability to make that protein. So if you have a genetic variant, typically it's in the gene body or maybe in the promoter sequence that stops this protein being produced, then it's going to have more severe impact in terms of pathology and human health, because that protein could be key in a particular molecular pathway or metabolic pathway. So understanding the impact of a mutation in terms of how it impacts on the protein is really important. And that's important when we're designing medicines, when we're trying to do precision medicine, when we're trying to understand what the impact is of a particular mutation or a particular treatment in terms of the desired protein we want to produce or we want to suppress if it's not a desired protein, or whether or not an abnormal protein is being made that we want to prevent being made. In reality, transcription is way more complicated, so this slide just adds a little bit more complexity. So here we've got that coding region of the gene, this is where that ATG would be, and this area here is the core promoter with key recognition sequence. As you can see here, the DNA preceding the gene that's being transcribed and translated stretches all back and coils around, and then there are areas way upstream that are really important in regulating if this gene gets switched on or off. And key areas in the DNA will be enhancers and silencers, where other proteins will bind and enhance transcription, so to encourage transcription and speed it up, or to silence transcription to suppress it because that protein is no longer needed. And through this complex interaction between transcription factors, activators, repressors, enhancers, is the very sensitive and highly complex way in which our body is fine-tuned to produce proteins in a homeostatic mechanism in response to particular stimuli, as well as doing all of this during embryogenesis as well. So again, what causes mutation? Mutations can just happen through errors so every cell of your body's got to copy all of that information all the time. Errors happen if the cell doesn't pick them up and apoptose, and they can be accumulated. And sometimes de novo mutations occur or through exposure to mutagens. So the impact of mutation, as I mentioned, is often really determined by where that mutation has an impact in terms of the protein structure. So does it change an amino acid? So here we have a protein, and obviously proteins are made up of strings of amino acid. If a mutation can change that sequence of amino acid changes, is likely to change the protein structure. If it disrupts completely that sequence of amino acids and doesn't produce a protein, it's likely to have a really severe effect. So again, understanding our amino acids, and this is a table of all the amino acids we have that make us us, and that our proteins are made of different combinations of these amino acids in different orders, and understanding that mutation has its effect by changing the amino acids or the protein that is meant to be coded for by that piece of DNA. That's really important, understanding the impact of mutation on the protein. So I mentioned complex disease at the start, and I think it's really just important to understand that we can focus on genomic medicine, and understanding genomic medicine is really important because it helps us to understand underlying pathogenesis of particular conditions, whether they are inherited conditions or whether they're somatic mutations that a cancer requires. It can help us to understand the pathogenesis of the condition. But also we must understand that for most complex diseases, it's really hard to delineate the difference between hereditary components and environmental components. And I've included heart disease here as an example of a complex disease, but this is equally applicable to diabetes, schizophrenia, cancer, high blood pressure, and Alzheimer's disease. How do we know if something has a genetic risk? There are thousands of papers for all of these conditions on identifying SNPs or key genetic variants. And there is undoubtedly going to be genetic risk for all of these conditions, but equally there's going to be a huge environmental risk, and the answer is we just don't know how much is environmental and how much is genetic for the majority of complex conditions. Twin studies give us some hint towards what is genetic, so if a condition is genetic or has a stronger genetic component, you would expect concordance or similarity between identical twins. And for most of the complex diseases, the concordance is pretty low, reflecting the important influence of environmental factors. So looking at heart disease, I've put a column here of elements that could be determined by your genetics, hereditary determined components, but equally very similar aspects that can determine risk, that could be risk factors for heart disease that are environmentally determined. So, and it's often very difficult to separate out the two. So I think this is an important thing to think about when we're looking at genetic studies and trying to design treatments focusing on simple genetics, that for most complex diseases there are multiple factors that will contribute to that pathogenesis, both environmental and genetic. And this is why epigenomics is particularly interesting. So with epigenomics, we're not looking at heritable differences. So essentially with epigenomics, we're not looking at mutations or variations in DNA. What you're looking at is changes in DNA, in gene expression. And there are elements of epigenomics that are fixed, but there are also elements of epigenomics that are transient. So it's one of the fine-tuned ways that our body appears to undertake homeostatic control, in that our body can respond to environmental stimuli. So environmental factors such as toxins, our diet, stress, exercise, smoking, alcohol, drugs, pathogen, and weather will all impact on our physical phenotype and health. And the way in which they do that is through likely to be through epigenomic mechanisms. And these are not changes to our DNA, but changes to gene expression that are on the surface of the DNA that allow a gene to be switched on or off to varying extents. So we have DNA methylation, which is a most studied epigenomic mechanism whereby methyl groups are found to be added in the promoter region. And when these methyl groups are found in the promoter region, they typically suppress a gene. So they stop a gene from being switched on or decrease the amount that a gene is switched on. So this is one mechanism whereby that level of protein that gene codes for can be controlled in simplistic terms. Histone modifications and chromatin remodeling is another epigenomic mechanism, and this is whereby how accessible that piece of DNA is made. So in order for a gene to be switched on, it needs to be accessible to all of the transcription machinery. So it needs to be uncoiled and not coiled around the histone proteins that make up the chromatin structure. So if a particular gene on the DNA and the DNA is tightly coiled around histones, it won't be accessible to that transcriptional machinery, so it's unlikely to be switched on. So the uncoiling, the remodeling of chromatin, modifications to histones can make a piece of DNA accessible or inaccessible to be switched on and to be controlled to produce this protein. The mice are always given as a nice visual example of epigenomics. So these two mice were the two offspring of two different experiments. So their mothers were cloned, so genetically identical. One of the mothers was fed a diet that was high in bisphenol A, which is a toxin. And the toxin is known to strip off methyl groups. And so all her pups, so the mum was brown herself, but all her pups were yellow. They were agouti mice because this particular gene that controls pigmentation and susceptibility to obesity in the mice was now expressed, whereas normally it wouldn't be. It would normally be suppressed. So the brown mum had all these yellow pups that all became fat. The same experiment with the other brown mouse, they also gave her the poison, the BPA, but they gave her a diet that was rich in methyl groups, methyl donors, so things like folic acid and betaine and vitamin B12 and choline are all rich in methyl groups. And this diet was sufficient to counteract the BPA that the mother was given. And so all her pups were brown, and they didn't have the agouti phenotype, they didn't have the stripping of the methyl groups. So this is kind of given as a quite a powerful illustration of how a physical phenotype can be modified by environmental factors. And this is really important to remember, particularly when we're referring to lifestyle advice, which is arguably going to be still the most important factor for improving people's health in terms of lots of the complex diseases we're facing. I'm going to go through a few slides just illustrating a bit more detail what epigenetics looks like. But another key example of why epigenetics is important is that fact that in every cell of your body you have your same DNA, but you don't want cells in your brain developing into liver cells, and you don't want cells in your liver developing into neurons. So a key way in which genes are switched on or off specific to where they're going to differentiate is through epigenetic mechanisms. So for example, the gene that's important for differentiating a cell into a neuronal type of cell will be hidden and suppressed in the liver, but it'll be nice and active so it can get switched on in the brain where you want it to differentiate into neurons. And so it's effectively what epigenetic mechanisms do just change whether or how much of a gene can be transcribed into RNA. And so the methylation can occur on histones in terms of changing that histone modification, but also the methylation can occur directly on the DNA and affect the DNA as well. There are some mechanisms that can cause permanent changes in expression. There are some mechanisms that are transient. This is just illustrating where methylation occurs. It occurs between in cytosine-rich areas, which are typically the promoter regions of genes. So when we look at mechanisms exploring methylation on DNA, we typically look at CPG regions for differences in methyl groups to indicate whether a gene is going to be likely to be suppressed or activated. And this is just showing some of the modifications to histones, whereby essentially all you're doing is uncoiling the DNA from those histone complexes to allow it to be accessible to promoters and transcription factors that are important in switching that gene on. So if we look at it on a global picture, we've got your chromosome, which is effectively chromatin, which is DNA coiled around these histone proteins, and the DNA. So in your 46 chromosomes, you contain all your DNA, half from mum, half from dad, and that comprises about 20 to 30,000 genes that determine everything about being a human and maintenance and living as a human being. And the ways in which those genes are controlled and differentiated is through these epigenetic mechanisms, which can be methyl groups to the surface of the DNA, or it can be through histone modifications of allowing areas of the DNA to be more or less accessible. So why is this all interesting? From the perspective of personalized medicine, understanding genetics at a genomic level, so understanding multiple genetic variations, understanding impact on protein function, understanding how a tumor may behave, so understanding the particular mutations with somatic mutations within a cancer can help us to design specific treatments that will or won't work for a patient. So that can all be really important. The other important thing is that your DNA doesn't necessarily hold your destiny. So just because a person has inherited a genetic variant doesn't always mean that that is the only way in which we can treat a condition. There are other mechanisms now we can focus on epigenomics to understand how we can increase expression of a protein or suppress a protein or design a drug that might help encourage expression of particular genes to produce a protein that is needed. Another key concept that's really interesting that we can touch on later is the concept of transgenerational epigenetic inheritance, and this is the idea that in conditions that are particularly severe or harsh, particularly stressful, or involve things like famine, the impact of that environmental factor can lead a lasting epigenetic imprint that can be transmitted to subsequent generations. So is the way I live my life and things I do and my diet likely to impact on my offspring? So whereas before when we focused just on genomics, we would say no, you are what your genetics determines you are. But what we're realizing now that there's also that importance of environmental contribution, both for the individual, but both in terms of the legacy of what they inherited experience from their ancestors. So this has really interesting potential for discussion in terms of how we understand risk and disease risk for future generations. So lots to think of. What I would encourage you to do now is to work through your genetics workbook and maybe start to think about what condition you find particularly interesting, if there's a particular disease, if there's a particular concept, because throughout this course we want you to encourage you to develop your own particular interest in genomic health and medicine, whether or not it's along the epigenomic line, whether it's on looking at RNA or DNA, looking at drug design or looking at a specific disease. We're happy to help develop that interest towards your project at the end of the module. Thank you.